Hi, my name is Tiffany Aguirre, and I am presenting a poster entitled Evaluating Cognitive Deficits as a Non-Motor Symptom in Parkinson's Disease Using a 6-Hydroxydopamine Lesion in the Substantia nigra. So, though we don't know the etiology of Parkinson's disease, we do know it affects nearly 1 million people a year in the US. We also know it targets the nigrostriatal pathway, specifically dopamine in the substantia nigra, both of which are crucial to motor movements, which is why we see the rigidity, tremors, and slowness in movements called motor symptoms. However, over the years, more patients have been reporting issues with pain, mood, sleep, and cognitive abilities, deemed non motor symptoms. Subsequently, dopamine and the substantia nigra are also key in reward and motivation, which is why this project is aiming to find if depleting dopamine within the substantia nigra would affect cognitive abilities, specifically decision making, by using a well known model for Parkinson's disease by injecting 6-hydroxydopamine, a neurotoxin that targets and kills dopamine, in the substantia nigra of rodents. Decision making will be assessed by a novel version of the rat gambling task at 2 and 4 weeks, as 4 weeks is supposed to be the maximum amount of dopamine depletion. It was hypothesized that animals injected with 6-hydroxydopamine will perform worse on the rat gambling test. Specifically, animals tested at 4 weeks will do the worst, as that should be maximum dopamine depletion. We used 40 Sprague Dolly rats, and they were food restricted until they reached 85% of their body weight to encourage reward seeking behaviors. After, they were taught to single and dual lever press via operant training. Then they learned our lab's version of the rat gambling task, in which one lever is deemed the advantageous decision and gives out a consistent but small reward, 1 pellet, 90% of the time, whereas the other 10% of the time induces a 10-second timeout with no pellet reward. The other lever is deemed the disadvantageous decision and gives out a large but inconsistent reward, which is 3 pellets, 40% of the time, whereas the other 60% of the time, a 40-second timeout is issued with no pellet reward. Then they underwent standard stereotaxic surgery, in which 20 animals were unilaterally injected with 6-hydroxydopamine in the substantia nigra, and the other 20 were injected with saline. After 2 weeks, 10 from the 6-hydroxydopamine injected group and 10 from the saline group were tested using the rat gambling task to assess any decision making impairments. After testing, animals underwent a transcardial perfusion to fix brain tissue and then immunohistochemistry to stain for tyrosine hydroxylase to account for dopamine, or lack thereof. The same procedure was repeated for the remaining animals at 4 weeks. Results found that there was a significant difference found in percent accuracy in that animals injected with 6-hydroxydopamine tested at 2 weeks had significantly lower scores than both saline groups. Surprisingly, animals injected with 6-hydroxydopamine tested at 4 weeks were not significantly different from any of the groups. This could potentially indicate any type of adaptation or compensation mechanism that could happen over time. So, even though our hypothesis was partially supported, we believe we may have found a potential link between decision making and Parkinson's and the role that depleting dopamine might play. Of course, this is the most basic biological aspect of decision making. As we know, decision making is an extremely complex mechanism that incorporates different brain regions, such as the hippocampus and the prefrontal cortex, as well as learning, memory, and even emotion. This is why I believe it is imperative to continue research by looking at depleting dopamine in other brain regions that are affected by Parkinson's. Thank you.